All right. With today's lecture, I'm going to start talking about concepts that will apply to our annealing of cartridge brass lab, but I also want to start our discussions of some of the writing assignments, and particularly I'm going to talk about the lab reports. So I'll do that first. But before we get started, I just want us to think about, some of the assignments and so forth coming up. Your your first lab assignment, the pre-lab. You've been working on it this week. Remember, that is due at the end of this week, so Sunday at 11:59. That's when a lot of the assignments are going to be due. But you can always check Canvas to see when those dates are. We will start data collection for this first lab this coming week. Data will be posted as we get it. Videos will be posted at the beginning of the week so you can look at the procedure that we're following. And then you'll have the worksheet for week one due next Sunday. Coming up beyond that, the second lab assignment, which I'll have posted to you soon, is going to be due in a couple weeks by Friday, September 11th at 5 p.m. So those typically are due at 5 p.m. But again, always check on Canvas for those due dates in case they change. And if you have any questions, let me know. All right, so now let's talk about your writing assignments and particularly your lab reports. So as you know from the syllabus, we're going to have two lab reports, a longer lab report on this first lab, brass, and then we are going to have a short lab report for the polymers lab, which is number 3. The other two are going to have a different form of writing assignment, and we'll talk about those on a later date. But let's get started on the lab reports. So I'm here in the course website on Canvas. If you go to modules, I put an extra module at the end that says writing assignments, and so this is just all the files that are going to be relevant to those. You can also find this information in files in a similar name, but I just thought I'd put it in the modules as well. But I want to draw your attention to the lab report guidelines. So this is kind of the master document for all your sort of information about formatting, page limits, all that sort of thing. And so I just thought we'd go through this real quick. There's some information about lab report notebooks. You can read through that, but we won't focus on it. So lab report formats, and I've separated them into full and a short. So this one is the full laboratory report. So this is what the brass lab is going to be. From MSE 202, this may not be as full as that. If you're familiar with MSE 407, this is the exact same format as that class. But briefly, the main things are the page limits here. You'll have a title page and abstract, that's just one page. Introduction, just one page. Theory and background, up to 3. Procedures, up to 2. Results and discussion, this should be the focus of your report. This is 6 pages, and then 1 page to conclude. And then figures and tables, again, if you were in MSE 407, you're familiar with this format that I have where the figures and tables are all sort of dumped in this section that's after the conclusions. And again, the reason I do this is I think it makes the formatting in Microsoft Word and other things go much easier. So those are all separated out, and you've got 4 pages for that. I don't put any caps on references and appendices. I typically recommend against, but if you need to put that, those are not capped either. The title page is formatted. You can kind of see the general format here with the title, which should be descriptive, your name, UK, and materials. The lab, that should be MSE 408, and the materials engineering 2. The date, and then the abstract there. If you don't do the box around the abstract, that's fine. Don't worry about that. All right, let's go over the various components that you're going to put in the lab report, starting with the abstract. So the abstract is going to be a short section, 200 words or less, and basically you want to communicate enough information so that somebody knows what's in your report and hopefully then reads your whole report. And so I kind of phrase it as a series of questions that should be answered in the abstract. First of all, why did the authors do what they did? So, what was their motivation? What information are they trying to give to the community, or what problem are they trying to solve? Um, so, after you do that, you want to say what you did. So what was the actual information, the results? How did you do it? So you briefly want to sort of show people the procedures. So you don't have to go into a lot of detail here, but you might just say you use TEM to get images, something like that, very quick. And then you just give some conclusions. So we kind of frame it as those series of questions, but it should be very quick. It should have results, so you shouldn't leave it as a suspense. Leave it suspenseful for the reader. They should find out what the results of the experiment are. You don't have to hide that, but you shouldn't cite figures and tables. That's usually not appropriate. The introduction. Again, this was only a page, so this isn't a very long section. Basically, the overall purpose here is to again establish the importance, motivation, tie it to problems, maybe in industry or in research, just lack of knowledge, what this is giving us, and then give your hypotheses for the experiment, and then the methods that you're going to use to test that hypothesis. So it should be pretty short, just motivation, introduction, and then how you're going to, what the hypotheses are and how you're going to address them. So pretty short section. Theory and background. So this will appear in a long or full lab report, and this is to give background on the various parts of the experiment that you're doing. So you don't have to go over stuff that's not covered in the lab report, but just enough to kind of understand the topics that are covered in your lab report. And so with everything, it's always useful to know the audience. So in this case, you're not trying to catch me up on theory and background. Your audience should be a student coming into the MSE 407, 408 sequence. So they have some of the sort of fundamental MSE classes, but they haven't done this lab. So that's kind of, and that's the audience I'll have you use throughout this class as well. So if you're ever wondering who the audience is, think of it as an incoming junior. So backgrounds covering those things. Also covers equations that you may use in your analysis. So those should be put in there. And I've put a sample format here. So your equation centered, and then the number to the right. And then you want to give the explanation of the equation with all the variables. So that should be in the theory and background. So just as a heads up, the theory and background we'll have in this long lab report, but this section gets cut out in the short form because I'm assuming as you're writing this that I can infer what the background is, or I can understand that you know the background based on your writing. All right. Next, experimental procedures. This is all the stuff, and so in this section, this semester in particular, this is going to be a little different, obviously, but this is going to be all the equipment, materials, procedures that were used, not necessarily by you hands-on, but you're going to write it as if you and your group did this experiment yourselves. So, you don't have to frame it as, we're doing it for you. You're going to write this as if, as a class, we did this. So, don't try to add anything about that. That isn't going to be necessary. But you just want to briefly communicate those procedures. So you don't need to give somebody an operations manual on how to use, for example, the rolling mill. You just need to tell them the conditions on how you did it and what piece of equipment you used, and the materials that you used. that's the kind of level of details that we're going for. And we have an upcoming assignment on procedures, and we also have a writing assignment on operations manual, because I really want you to sort of see the differences between those two types of experiments. Results and discussion. This is by and away the most important part of your lab report. So this should, if you're going to skimp, don't skimp on the results. This is where you present your results and your analysis. What do these results mean? This is where you're going to present the figures and tables that are in a separate section, but you want to make sure that you're using this section and you're talking extensively and drawing on external resources to compare your results and see if they're valid. So this is going to be a major part of your experiment. Before we get to figures, there's a conclusion section. This is, again, just a kind of wrap up. What were the major findings? Did it address your hypotheses? You can talk about error here as well. So just to kind of wrap up, and sometimes you can even mention what sort of future steps you could have in this experiment. Okay, so figures and tables, like I said, that's split off. And so these are going to be in this section, and they're going to be in the order in which they appear in the text. So as you're writing in the results section, or even procedures, however it appears, whatever comes first, right? So if figure 1 is the first thing, if a figure of hardness is the first thing that you reference in your lab report, that will be figure 1 and appear first in this figures and tables section. If a table is the first thing, then it would be table 1, and you'd give it a name, and that would appear first. So it's in order that it appears in the text. So that's a very important part to include. References, obviously this is where you should have a few references. I mean, I think that's usually, if I see 1 or 2, that doesn't seem like an appropriate level for this type of report. So you want to make sure that you're comparing and contrasting the data that you have. So this often takes multiple references in itself, but then you also have background information. So particularly when you have theory and background, there should be quite a few references. But again, there's no minimum, or there's no maximum. So I'm not counting this in terms of page limit, but make sure you have an appropriate level of references. It's going to be in the IEEE format. I've put some PDFs on the module and also in the files about the IEEE if you're not familiar with that usage. You're welcome to use something like EndNote or Pages or any other kind of reference maintenance software, but you don't have to. So that's also an option for that. Again, references are particularly important. If you fail to reference something, that is plagiarism. We use the Turnitin website, and so it'll catch things like that. So you want to make sure that you're using the references to compare, but also if you're saying something in your report, you want to make sure you give proper credit to those references. Appendix, I won't really go over that. Usually this is a case where if you think you need an appendix or something, you should probably kind of chat with me and see, but it's kind of stuff that shouldn't be in the body of the report. It's not just a place there so that you can save on page limits. Okay, so that's the full lab report. Let's briefly look at the short form. So this is the purpose of the short form is obviously it's going to be shorter, but the idea behind this is similar to if you've ever read an academic journal letter, that format. It's really sparse, and it really just goes into the results and so forth, and it's very short letter. So that's kind of the idea here. So a lot of people like this, some people don't. But the main idea is that you're trying to communicate a lot of information, but do it in a lot less space. So in my mind, these can be pretty tricky because you have page limits that are pretty severe, but you still want to get across all the information that you need to. So the title page and abstracts the same. One page for procedures, so that's half. There is no theory and background, so anything that you think really needs to be there should be in the results and conclusions. So that's down to 3 pages with both of those sections. And then you're going to have 2 pages for figures and tables. People really struggle with this, so make sure that you're thinking about how you present your data in figures, tables, whatever it may be. And then references, again, I don't put page limits on that. So everything else is going to be pretty similar to the full lab report. The main difference is the results and conclusion. This basically is almost all the information of the lab report besides procedures. So basically everything goes in here. So you're writing it as a continuous section. And so if you have theory that you need to get across, it should be short, but you do it here. Yeah, and you're just writing it so that the reader can understand the results, and then you end, with a brief conclusion. But you still need to have hypotheses and so forth. But again, it's very, very condensed. So this will be the short form will be for the polymer lab that we have third. All right, so that's the main thing I wanted to kind of go over is just a brief introduction to those 2 formats that we're going to write. Some additional guidelines are below. So information about, what type of font and the fact that it should be double spaced, all that. That information's here with section helps on section and so forth. Table information, figure information is here as well. We have some assignments coming up on figures, so hope to have some time to spend on what makes a good figure and so forth. But some of that additional feature can be found here generically for both of those assignments. All right, now that we've talked about some of the information about the lab reports, let's go over some of the basics of this brass lab. I don't go in an extreme level of detail here. I know you've seen this in at least MSE 201 to some extent, and then potentially a class like metals. So I'm just going to kind of give you the basic overview, and then I've given you some reference material if you need further information about this lab. But the first thing I want to show us is from the metals textbook, and it goes over the stages of annealing. And so I wanted to show this because, again, this is annealing of cartridge brass. The goal of this lab that I want you to obtain is looking at the stages of annealing with the techniques that we're using. So metallography, hardness, and conductivity. All right. And so this graph up here shows, this is actually taken from calorimetry. So it's energy released as a function of time at a specific temperature and reduction, right? And so it shows a release of energy at a certain time, right? So that's kind of corollary to the experiment that you're doing, because you're doing everything at one temperature and then looking at different times. So kind of this shows you what's happening. There's a little bit of energy released initially, and then there's this peak, right? And so it's kind of what's happening in those 2 regions is what we want to look at. And down here, I put this because this has hardness and resistivity, or same thing as 1 over conductivity. So those are 2 parameters we're looking at. And so this can be important. In this case, it's looking at as a function of temperature instead of time, but we can draw some comparisons to what we see here. And then down here, it's power difference. So this is very similar to the energy released that we have. So we're not doing this type of data collection, but this helps us understand what's going on. So hardness as a function of temperature and time can often have this shape. Resistivity, same thing, can have this kind of general shape. So hardness, you see that it's more or less flat, and then it has a steep decrease, and then it's flat again. That's the general kind of trend that we see. Resistivity, pretty similar. It has a steep increase where hardness decreased resistivity decreased at the same time hardness did, but there's also this additional decrease at this lower temperature. And the reason it shows this down here is because where resistivity initially decreases, we see a peak on this power difference. So this is a DSC, which we're going to run later in the semester on polymers. And then a larger peak where both hardness and resistivity decreased. And so it's basically telling us information about the physical state of this material. So if this were happening to our material, I would expect that this region at around 600 degrees was recrystallization, the formation of new grains from highly distorted grains, right? So we have a huge release of energy, hardness decreases, resistivity decreases. So we have heavily deformed grains, so lots of plastic deformation, and so hardness would initially be high and then decrease when we form new very low, annealed grains that have low amounts of disorder. And same thing resistivity, there's less resistance to the flow of electrons. And then there's a power release because this is an exothermic event. So that's recrystallization. So what this kind of shows us is that all three of these techniques would be valid for determining where recrystallization is. And we should also be able to see it in the microstructures, which we'll look at in a second. But you'll see that this initial blip here that also corresponds to resistivity, you can't see on hardness. So if you think about what this is, it's not recrystallization, it happens before. So that's going to be recovery. And so recovery cannot be identified by hardness because these structures are changing in such a way that you don't see this on a bulk level until you get to recrystallization. But you do see it in terms of resistivity in some cases because electrons would flow through a material can flow faster if there's less resistance on a very small length scale, right? So on the order of dislocations moving around, right? So if dislocations were moving around in recovery such that they're forming lower energy structures, then electrons would be able to move around them on this microstructure. And so you can sometimes see recovery with resistivity or conductivity, which is why we're using that. And also, if you're using something like DSC, we can see that. So that's kind of the power of these techniques, and you can kind of understand why we're doing the techniques that we are in this lab. All right, so let's check to the other, let's go to the other part of this experiment, which is the microstructures. All right, so this is cartridge brass. This is taken from a previous semester. And the two things that we want to look at are cold work, the effect that cold work has on cartridge brass. And we also want to look at the effects of annealing. That's the goal of the experiment. So in this structure, you can tell that all of these grains are very long, right? You can see these long grains running top to bottom, right? That's one thing. You also see a lot of these hash marks, right? These are telltale signs of deformation, particularly rolling or some form of very isotropic deformation. So rolling is basically squishing those grains so that they run parallel to the rolling direction. So that's our rolling direction. And these hash marks have to do with deformation. So we'll talk about that too. This is just a zoomed in version of that to kind of show you the various hash marks and the rolling direction. Same thing, even larger view. All right, here's another view. Still cartridge brass, same material we've been working on, but this is clearly different, right? Overall, you see that these grains, so for example, this grain, is more or less what we call equiaxed. So same size in all directions. So roughly, it's not going to be circular because of the different crystal facets, but it is roughly the same in all directions. So that's one feature, that most of these are roughly equal in all directions. They're fairly large grains. The other thing you noticed is these parallel lines within a grain. These are known as annealing twins. So this is something that hasn't gone through cold work, so it hasn't been rolled, or it's been rolled and then annealed such that we have this structure again. So this is kind of our baseline structure before or after the cold working and annealing process. But it's annealed grains that are equiaxed. This is, again, same material, taken on a different microscope, which is why you have kind of the different tinting, but it's the same material. I just wanted to show you that sometimes it appears in different shadings and so forth. The color doesn't really matter that much. All right, here's another image. So this looks similar to the first one I showed you. You've got these long grains, but one thing you'll notice is all of this very small features that we see. All these, it almost looks like confetti. These are new grains forming. So this is something that's been cold rolled, right? Rolling, we can identify a rolling direction, but it's undergoing the annealing process, and particularly recrystallization, right? Because we've crystallized these new annealed grains that are very small initially. So all of these little confetti pieces all around are annealed new crystals. So it's undergone recrystallization to some extent, but we haven't fully recrystallized because we can still see some of the grains that are unchanged. This is further along than the last one, so we really can't identify a rolling direction, but the grains are really small. So this would tell me that this has probably been cold worked, rolled, and maybe be able to tell the rolling direction is vertical, but it's hard to tell. I couldn't be confident in that, but it looks like it's almost all recrystallized, or it is completely recrystallized. So it's right at the end of recrystallization, or it is the end of recrystallization. Here's another one. This is, again, rolling. You can see the rolling direction, these really long grains. But I showed this because this is a really high magnification on light microscope, but you can see these little grains. It's a really nice image because you can start to see the new grains forming. So this is right at the beginning of recrystallization. So this is kind of a warning for you that once you, when you're looking at your microscope images, or you're collecting them yourselves, if you're looking for recrystallization, make sure you look really hard and make sure you look at the highest magnification to see if you can see some of these new grains forming, and also pay attention to where they're growing. They tend to grow in specific sites in the grain structure, so really pay attention to that. Same thing here. This is really clear that the new grains that you can kind of see popping up are growing in particular features. So I'm just going to highlight the fact that they're growing in these particular regions. So I want you to think about that, where they're growing and what that means. Again, I'm just zooming in here so you can see that. All right, so this is again from the metals textbook, showing you the overall, kind of the overall process of annealing through micrographs. So rolled, annealed, starting to anneal, so you might see small grains. Those grains are growing, and eventually they consume your prior grains. And then at the end, the process never really stops, because that last process, as you're aware, is grain growth. And so grains can grow indefinitely. And so that's why you'll see that these continue to grow and grow and grow. So that's kind of the overall stages of annealing, and that's what we're going to be looking for through hardness, conductivity, and with our microstructures. So you want to have a really good understanding of what you're looking for this experiment. Again, everything here was framed in terms of increasing temperature. So it's going to be a little different now that we're looking at isotherm and then changing the time, but a lot of the principles still apply in terms of what we're looking for in annealing.